Hello, everybody. My name is Dr. Nikos Xynos, and for the next about half an hour, you are going to be watching my virtual presentation regarding cannabis-based herbal medicines and bioprocessing, which is delivered in the frame of the virtual course organized by the National University of Colombia and the Chemical and Biochemical Processes Research Group. I'm going to share my screen with you and start presenting my topic. So I'm going to talk to you about cannabis-based herbal medicinal products development, which in fact is pretty much a journey into botanical bioprocessing. Again, my name is Dr. Nikos Xynos. I am a science and technology partner working with MedColCanna in Colombia. And I would like to make a short introduction. So Hippocrates used to say, let food be your medicine and medicine be your food. And this could not apply more to cannabis, which delivers products that range from food to nutritional supplements and to medicines and prescribed drugs. The limits sometimes are blurred. Food can be medicine, but medicine and food can both be poison or poisonous. We know that last year, FDA issued several alerts and against companies that are producing CBD-based products, while at the same time, CBD cannabis oil producers are struggling to improve the quality assurance and to reduce the risk for the consumers. Sometimes FDA issues even warnings to companies for unsubstantiated health claims, and at the same time, within the same year, last year, FDA approved for the first time in history a drug which is directly purified from cannabis biomass. So let's look a little bit into the facts in order to start structuring this landscape. Cannabinoids. So cannabis-derived extracts contain more than 500 identified secondary metabolites. Cannabinoids alone have more than 100 different chemical structures. Chemically speaking, most of these compounds are terpenophenolics. This means that they are compounds of medium to relatively low polarity, so they have a relatively hydrophobic nature. This also depends whether we are talking of the acid or form. As we know, those are present within the plant, mostly in their acid form, but in the products that people consume for their health, they are found and stabilized in their neutral form. And of course, there are way more other classes of cannabis metabolites. There are more than 140 terpenes present in cannabis, and then there are other compounds like flavonoids, other phenols, sterols, triglycerides, etc., etc. So there is a richness of natural products into the cannabis plant, and more specifically into the flower and leaves that we used to consume or extract. So how we can structure cannabinoid-based product development to be safe and based on quality assurance principles. We have to come up with an efficient process that will be based on clinical trials or other bioactivity testing data. At the same time, we need to ensure that we have enough toxicological data and assessment. And of course, all this has to happen through a quality assurance system. It means that we need to dedicate budget, time, and know-how to research and development activities. Now, let's look first into efficacy, into cannabinoid efficacy. So, most people today are talking of CBD and THC, and most companies as well. And most products, either they're food or supplements or medicinal products, they focus on these compounds. They are the major metabolites, and they have drawn attention and clinical interest. There's also the so-called entourage effect, which means how well substantiated is the entourage effect is talking about the broad spectrum effect, the effect of the broad spectrum of the cannabis component into enhancing the activity, biological activity of CBD and THC. There is a lot of talking about it. There are efforts to prove its existence, its very existence, but yet we don't know how well substantiated is this and how this translates into efficient, safe, and quality products. And of course, there is an increasing interest in minor cannabinoids. Of course, we all know, most people know about, have heard of CBG or THCV or CBDV or CBL, CBC, so cannabidiol, tetrahydrocannabivarine, cannabichromene, etc. And those seem to have therapeutic potential. But again, closely, have we looked into this therapeutic potential and how well substantiated is this before going into developing? So the first steps of an industrial venture into the cannabis, the so-called cannabis space, which again can be focused on the food space, food industry, or supplements industry, or medicines industry. These industries are widely different and with very different characteristics. If we want to talk more generically, at least at the first level, then there has to be an adequate process and unit design that will guarantee that efficient and safe products will come out from this unit. So there are various steps of processing in order to produce products that derive from cannabis, and these can be, there can be various alternatives and steps. But if we want to summarize a little bit what exists today and what is, let's say, the standard of the industry, we will be talking, first of all, of first fruit extraction, then some processes of chemical transformation, along with further processes of enrichment, then fractionation, targeting more and more deeply into the cannabinoid content of the extracts, and going down to even purification of isolates, of cannabinoid isolates, that then need to be formulated into efficient products. So in order to have a structured research and process development, we need to look closely, first of all, start from product specifications. What is going to be our final product? In which jurisdiction is it mainly going to be looking into? And what are the regulatory constraints? Because there are constraints, as we all know, in the cannabis space when it comes to regulation. This is the first. So we have to start backwards. We have to start from what's the product, what's the final product, and then go back and see how we structure our research and process development. After this, we are going to be looking into our extraction technologies and solvents that we're going to be using. And of course, we are going to be more and more on polarity and efficiency of these solvents, but also the technology that we're utilizing and the pros and cons of its technology, based always on what we want to do as a final product. And we're going to look into the enrichment and purification step, the formulation and the packaging, and of course in how we implement and scale up, implement production based on research and process development. So we go from R&D to scale up our processes, to scale up our machinery, which technology suppliers should we select and work with, what's the scalability technology, and what's the return on investment finally. So let's start from the positioning of our products. So we need to define which one will be our active pharmaceutical ingredient, which cannabinoid. Is it going to be the delta-9 THC? Is it going to be the CBD? Is it going to be both of them? Or are we going to focus in minor cannabinoids? Or do we want all of them present in an extract or concentrate or distillate or whatever? And of course, then we have to look into the limitations. Which of the substances that are included in our finished product as an API, as an active ingredient, or as an impurity, which ones of them are scheduled in the jurisdictions that we're looking into? Is it 9-THC alone? Is it the CBN? Is it any other? And are those the target compounds, the APIs, or are those impurities that we need to characterize, that we need to profile? And what are the limits in the contents? And of course, this is related to the eventual market authorization for these products, so, which depends heavily on the authority that we're going to address our demand, our claims. So are we looking into prescribed drugs? Are we looking into delivering medical devices or supplements, nutritional supplements, functional foods, cosmetic products, or even some more specific categories like botanical drugs, etc.? The geography and the jurisdiction of our both production and distribution is very important. If it's in the European Union, for instance, the regulation and the rules are not homogeneous. Where are we looking? There is a frame, there is an umbrella. Where exactly are we looking into? Which country? And if it's North America, is it the US? Is it Canada? And then, if yes, which state of the US or any other country that can be in the world, either for producing or for delivering, for distributing these products. So then once we have the specifications of our finished products we desire to produce, then we have to go back and start from the first processing step. Looking first into the existing optimal extraction technologies and so I say optimal because there exist various extraction technologies and processes and solvents implicated in the botanical extraction space. And for cannabis, we have to focus. We have to start by focusing into those that are the most efficient. So the criteria indeed is efficiency and recovery, using as much as green and non-toxic solvents as possible. Then we have to look if we want to work in with a food or pharma grade machines and solvents. And we have to focus on the scale of our production, because this might be very important when we are going to be selecting our optimal technologies and processes related. Some processes and some technologies perform very well in pilot and small production scales, while other ones perform better and have higher interest, present higher interest in very large scales. So we have to define our scale of production, at least for starting our project. Then, of course, the field of application is very important. Are we going to be doing food or pharma products, etc.? And of course, all these come down to the capital expenses and the operational expenses of the unit. So looking closer into what extraction technologies and solvents are available and are the standard of the industry, so by definition, the optimal, we are going to see that these are various, although they are limited compared to what generally exists. Again, still there are various. So supercritical CO2 is a standard of the industry, utilized with or without co-solvent. Ethanol is usually a co-solvent that might be used in this case. So supercritical CO2 is a green solvent. It is non-toxic, non-flammable. It produces the least possible residues, of course, because all this mainly because it is in ambient temperature and pressure, it is a gas. So we're using it in a supercritical condition within the system, the extraction system, but then when we recover the extract, then there is no solvent practically. Then it has some special characteristics, such as it has a high capex but a relatively low opex, and the systems that exist are very well sophisticated. They are GMP compliant. Of course, they can also be very simple systems, but there exist very good systems, GMP compliant, with advanced controller data logging, with all the specifications, all the user requirements and specifications that need to be in place in order to have GMP compliant equipment, pharma GMP compliant equipment. But still, in the industry today, still exist pressurized hydrocarbons, such as propane and butane. Although there are questions about their flammability, they're flammable, about the GMP compliance of the systems that operate with these hydrocarbons. There is some progress compared to the old school garage systems that, let's say, started and boosted this industry in the very beginning when it still was mostly illegitimate. But yet this progress doesn't seem to be good enough. Then, of course, there are other alternatives into pressurized solvents. Again, more GMP than the pressurized hydrocarbons, such as the tetrafluoroethane systems, that is the so-called R134A. So this is a very safe solvent, a food grade solvent. But how GMP, how is the compliance, the compatibility with GMP rules of the existing systems? And of course, there's not only near critical extraction, there's also liquid solvent extractions, alcohols like ethanol. These alcohols are very efficient into recovering cannabinoids. They can be used either in ambient temperatures, not in high temperatures because these are very volatile solvents, but either ambient or chilling temperatures. So very, very cool, below zero temperatures, so that we can avoid the extraction of heavier compounds such as waxes and hydrocarbons, etc. So then there are alkanes like pentane, hexane, heptane. These alkanes extract the cannabinoids well, and in some cases they are very safe. Mostly pentane and heptane are classified between the most safe solvent classes from the ICH, the International Council for Harmonization. There are other types of extractions as well, utilizing edible oils directly with the plant material, like olive oil or hemp seed oil or sunflower oil, or paraffins and other petroleum-based solvents. And solvents is one thing, but through which technologies these solvents will become more efficient into extracting the secondary metabolites and especially the phytocannabinoids from the cannabis biomass, this is a very important thing. Which technology? So is it going to be maceration? Is it going to be percolation? Is it going to be pressurized liquid extraction or microwave-assisted enzymatic extractions, ultrasound based, etc., etc.? So there are multiple alternatives and variants, and you can find out in the market many technologies that are being proposed by the manufacturers, many technologies that processes that implicate these technologies have been patented, have been scientifically published, have been, in few words, have been proposed to the industry and have been presented as good or better or optimal. So one has to navigate wisely through this landscape and find the most efficient process and technology. And when I say efficient, this depends heavily on the finished product that we're looking to do. And of course, it's not only about technological and scientific efficiency for extraction, but it's also about financial efficacy. So there's a lot of articles and YouTube videos and fairs, but there's not a single best technology for extraction. So ranging from simple hydrocarbons to supercritical CO2 and other safer and more efficient solvents, we can find various alternatives and solutions. And then when we come up with the best solution for our manufacturing, then we have to look into the manufacturer, because there are various manufacturers for each technology. You can find systems from North America, from Europe, from China. These are basically the geographical zones where these systems are mostly produced, and of course the specifications, the quality, the design, and the prices are very different. So, but it's not only about technologies, it's also about process development. It's about developing efficient standard operating procedures, and these standard operating procedures, by being optimized, these dictate what is good equipment and what is not good equipment, because these standard operating procedures optimize SOPs, tell us, give us the parameters that we need to use in order to have good efficiency for our extraction. And for instance, we know that we have to reach pressures that are quite important in supercritical CO2 extraction in order to fully recover CBD or THC or other cannabinoids from our cannabis biomass. So, me personally, I would not for a supercritical CO2 system that cannot stand high pressures, or that does not have the ability, the flexibility to utilize co-solvent, both for process efficiency and for cleaning of the system, especially when a cleaning place protocol has to be put in place, and especially when it has to be put in place within a pharma environment. This is very important. So this is an example. There is scientific knowledge, public knowledge, and of course there are patents, but still there is public knowledge readily available for us to let us and to give us instructions and to instruct us how to choose the optimal equipment. What are the traps that we have to avoid? With what kinds of machinery we need to be looking into? First of all, in terms of process efficiency, before going into GMP-related matters. And so we need to redirect our eyes from marketing-driven communication into scientific literature at first, and of course also at the same time look into the existing intellectual and industrial property. We want to have a machine that can implement the best practices according to the literature, and that does not breach any existing patents. Once we have these characteristics, and once we have done our research into this domain, or once we have consulted some expert in the field, then we are way better readers of marketing-driven communication, such as brochures and presentations, etc., etc. And of course, going into fairs, discussing through email or teleconferences with the manufacturers and everything. So the extraction technology, looking more close to these extraction technologies, and to the supercritical CO2 extractions first. We know that there are several points that we need to focus on. We need to look very closely into, first of all, what I just mentioned before. Our system can stand high pressures, pressures that are optimal for cannabinoid extraction. And then how, if it has, it contains, it includes a closed solvent circuit with the pump and the tubing, and the ability to utilize co-solvent for both more efficient extraction and more efficient cleaning. We have to look closely into the pump capacity, because this can affect pretty much our recovery of cannabinoids or our batch duration. So we might be led into compromises because we built a system with poor pump capacity. We have to look into the system and how practical it is to load and unload the biomass and to recover the extract through the separators. There are various options and systems out in the market and manufacturers that have more or less innovated on this. So that was an example for a supercritical CO2 system that this applies to every extraction technology that we might be choosing. So if we opt for instance, primary ethanol extraction, again, similarly, we need to look closely on the equipment after having looked closely on what's the optimal processes. So, and we have to find equipment that complies, that can do these processes, that is able to perform well with this process. And then moving forward to further enrichment and purification, or the so-called refining of cannabinoids. We need enrichment tools, and there are various again. So winterization is an enrichment. It's a filter. We filter out these and insoluble, at least insoluble in very cold. It's followed by a filtration step. But there are other filtrations that are not associated necessarily with the winterization. And there are decantation techniques, technologies that can substitute filtration, might be more or less efficient. And we have to look also into the thermal stability, because this affects the efficiency. The main thing in the industry for further refining after winterization is the so-called molecular distillation or short path distillation. But there are alternatives. There are alternatives, and these include the countercurrent supercritical fluid or liquid-liquid extraction as an alternative to molecular distillation. Molecular distillation is more of a physical-chemical process. It's based on the differences between the boiling points of the various metabolites and compounds that are contained in the cannabis extract. So it can be to an extent, it can be selective, but there might be more selective processes that are well known into the botanical extraction industry today, but have not yet penetrated so much the cannabis industry. Of course, this might be more complicated approaches than molecular distillation, which is rather simple, but still quite copious. Other technologies include adsorption resins. Practically, most people don't do this, don't apply these technologies into their industrial processes. So, but it's a good alternative to molecular distillation, depending, of course, for whom and for which purpose, for which final product. And then going down to purification and isolation. Here it's very important to know what are we looking into. Are we looking into a CBD isolate? Are we looking into THC isolate or their acids? Are we looking into minor cannabinoids? Are we looking into extracts that have been, that THC has been removed out from these extracts? So this is very important. And actually, this is mostly what will lead us to choose between recrystallization, which is an efficient and relatively simple approach, low cost also, although quite copious, but it's low capex. So between recrystallization, we have to choose, and between chromatography. And then, of course, when it comes to chromatography, there are various technologies and alternatives. There is silica-based chromatography like flash, like MPLC, HPLC, and then even SFC, supercritical fluid chromatography, or liquid-liquid chromatography like CPC, centrifugal partition chromatography, or CCC, which are relatively niche alternatives to costly silica. But I would say not so niche, especially for cannabis. Especially in the cannabis space, these liquid-liquid approaches have been pretty much involved in the industry and have been proved at, I would say, up to one scale. And then there are question marks from a scale and up and higher. So the scale is also very important. So the technological variabilities are many, and again, there is not one single best setup. All these technologies that I mentioned before can apply very well into cannabis, and for all these technologies, there are various manufacturers. Probably many more for short path distillation than for CPC chromatography, but various for each one of these technologies. So the landscape is quite complex. But let's say that the fundamental dilemma is chromatography versus recrystallization. So, do we need to implicate high investment and complexity steps, technologies like chromatographic technologies, or are we satisfied with what the relatively simpler and low capex recrystallization processes can offer to us? So, to chromatograph or not to chromatograph? If to chromatograph, then should we work with silica-based or silica-free? So, should we work with both? Should we have a two-step process that can combine both technologies? Who will tell us? There are always alternatives in terms of technologies and then in terms of manufacturing technologies. And one has to be an expert in order to come up with a definite, let's say, conclusion and choice. Things are presented somehow in marketing-driven communication so that the manufacturers of extracts and final product can be helped to select. But the way to present things is not always very simplistic. Or the way to present things in a simplistic manner, it's not always the best, because, of course, manufacturers are looking into presenting their technologies in the best way. But in my opinion, you cannot calculate an ROI, a return on investment, from a brochure. First of all, because the data are not universal. Sometimes they're not even realistic. And when I say that they are not universal, it's because they do not apply to all types of applications or types of target compounds, targeted materials, materials, and of course, for all types of desirable finished products. So, and of course, there's a question of different strains with different contents of cannabinoids, etc. So, the efforts that are done by the manufacturers of the technologies to present, to provide data through their marketing communication, through their brochures and videos and presentations, whatever, are good and sometimes are very helpful for the industry. But one has to be an expert, and one has to have expertise and experience in these kind of technologies with cannabis or other botanicals. Actually, this is also very important in order to be able to assess this information and see which is the most practical technology, the most efficient technology, and which is the so-called the best chromatography system for me, for a specific company project, of course, because these things cannot be universal. And again, various manufacturers from all continents, or at least mainly from 3 continents. This is very important to see. Actually, it's very important to have a user requirement specifications when going towards a manufacturer to ask for a quote or an offer solution. Finally, once the extracts or concentrates or isolates have been produced, these have to be formulated and packaged to be distributed in the market, whichever is this market. So again, here the options are, there are many options. Market there are from the simpler hemp extract that contains CBD, and is supposed to not contain THC or to contain low THC in this content. This is a relatively simple solution in terms of formulation. Then to more complex products, such as the Epidiolex, for instance. This is a drug, a prescribed drug. It's an oral solution delivering a standard dose. Then there is Sativex, an oral mucosal spray. There are soft gels like Marinols. All these products are pharmaceutical products, and so they are obliged. The companies are obliged to guarantee very standard delivery of active ingredient. And of course, going into the recreational market, which is a very different market, of course, or the supplements market, whatever this is. There are all these vape pens or these droppers, etc., etc. So our criteria can be various depending where which market are we looking into, which kind of products are we looking into. Availability, market authorization issues, active substance properties and delivery, route form of administration, and related matters. And there's more than this. There are gummies. There are, when it comes to food and CBD, this the sky's the limit in terms of forms of products. Mostly focusing on drugs and either supplements or medicinal products. We as mentioned before, we see bottles with droppers for oral sublingual administration, oral solutions like Epidiolex, capsules and like Marinol, oral mucosal sprays like Sativex, solid forms, tablets, patches, and then of course cosmetic products, or to put it broadly, topical products. Might be cosmetic or therapeutical topical products. And of course, when it comes to consumer goods, beverages and functional foods, etc., then this is a whole different story. Once we have worked through our process flow diagram, when we have selected our optimal processes and defined our optimal processes, sourced the optimal equipment that will be able to comply with these processes, and sourced manufacturers actually for this optimal equipment, then we need to look at scale-related matters. We need to look into bringing all this into production scale level, and of course, scalability of the processes is very important. Things can vary a lot when we do the scale up from lab to pilot and to production scale. The reliability of which technology can be different for scale, and even the feasibility of the project can be affected. And then there is a key point is the reliability of the technology manufacturers that we need to look into. So we need GMP compliant equipment for some units, or for others we don't need this. So we have to define what kind of GMP compliance. Food, this is very different. What other compliances, what other regulations and limitations we have to look into explosion-proof material? Either it is ADEX in Europe or X-proof in the US, or are we looking into Chinese equipment with China X-proof certificates? So this is important. Actually, we need to know the manufacturer of the products that will buy the machinery needs to know what will be its classification in terms of hazardous area, and then ask the manufacturer of the equipment to be compliant. It doesn't go the other way around, as often is seen in this industry that is trying to mature very quickly. So, and of course, we need to have a customized design for this is very important. We need to work with a manufacturer that will customize their design for us. That will be flexible, that will put some engineering know-how in order to provide optimal customized solutions for our facility. Another question is the know-how transfer. Usually I give an example to people that are looking into buying machinery and working with the manufacturer, that in the pharma industry, when you buy a bioreactor where you can realize a chemical process like a synthesis, etc., you buy the reactor, you don't buy the process, you don't buy the synthesis method, right? So in the cannabis industry, again, which is an industry that tries to evolve very quickly, so people need to accelerate the R&D in order to go into production and produce products. The manufacturers often provide solutions in terms of efficient methods and processes based on their equipment. And this is very important, as well as the training and the support. So everybody needs to work with reliable manufacturers that can provide methods and processes, or alternatively have a very strong R&D in-house, so that this R&D team, R&D and industrialization team, will develop the most efficient processes based on equipment that comes from specific manufacturers that do not provide efficient methods. There are manufacturers that still provide training and support, but training on the use of the machines, not the processes customized and optimized for cannabis, because their systems go to all types of industrial applications, not just cannabis. So there are different approaches. And of course, the after sales issue is very important. The services, the warranty, the responsiveness, the remote control, the level of remote control, or even on-site assistance is very important. And of course, geographical distribution. Sometimes communications is very facilitated by good representatives and distributors, and the pricing can vary a lot based on that or not. And then the terms and conditions that apply specifically for specific regions. So one has to look into these matters as well. So one has to navigate wisely to the destination because this journey into cannabis bioprocessing is an odyssey. So like Ulysses needed to navigate wisely to his destination, Ithaca, we have, first of all, to know what is our Ithaca, what is our destination, where we're going into. And then we need to come through the simple gathers, the sirens, and every other kind of distraction. And of course, we need to trust our partners in order to realize this mission, this complex mission. I would like to thank you very much for your attention. And I hope that in the future I can answer to any eventual questions that you might have through email, since I'm delivering this course and in a non-real time. Again, I'm Dr. Nikos Xynos. I'm a science and technology partner working with MedColCanna in Colombia. And thank you very much for watching this video. I'm open to your emails with questions and propositions and whatever this might be. Thank you very much.